In this seminar, we are going to have a discussion about scope of bioinformatics in Pakistan. We are going to have a discussion about career opportunities in Pakistan and specifically what you can do in bioinformatics to further excel in your career. The topics that we are going to discuss are introduction to bioinformatics for those who are not from bioinformatics background and from some other fields of bioinformatics such as biotechnology, microbiology, or any other similar fields. Then we are going to have a discussion about subfields of bioinformatics, what sort of advanced techniques are there and methodologies are there within bioinformatics that can help you to excel in your career. Then we are going to have a discussion about applications of bioinformatics in actual real life industry research and where you can apply your newly learned skill related to the advanced techniques and methodologies. Finally, we are going to have a future outlooks and challenges discussion. In this specific seminar, we are not going to have a discussion on the Q&A session because that was only available during the live session of this particular seminar. So I'm Vakar Hanif and I'm founder and CEO of BioCode. I'm a bioinformatics scientist, data scientist, and a computational researcher. I develop pipelines, softwares, and other tools for bioinformatics analysis that revolve around cancer genomics, molecular oncology, including drug discovery and other genomics projects and similar techniques that allow us to do cancer genomics projects. So it is one of my key research areas. I utilize various different NGS based datasets related to cancer genomics to identify new therapeutic targets for drug discovery. I identify new drug targets for both bacterial and human species, and I try to merge both cancer genomics with other new fields such as single cell genomics and metagenomics. So these are my key research areas, and I do get into other research areas such as applying drug discovery or NGS based techniques on diabetes or other similar diseases that are wreaking havoc all around the world, and I try and find new drug compounds, new therapeutic targets using RNA sequencing analysis, differential gene expression analysis, single cell genomics, single cell RNA sequencing analysis, and that is a basic background about me. You can learn more on LinkedIn if you are interested, and if you are interested in to learn more or get in touch with me. So let's have a discussion about introduction to bioinformatics. So basically, you might have an idea what is bioinformatics, and you might have taken a definition or read some definition from some paper, from some book. I'm not going to bore you with that basic definition. I'm going to give you exact reason where bioinformatics helps us in this day and age. So basically, bioinformatics is a field that helps us to analyze genome and its structure. So basically, each individual has a genome, and that genome can be sequenced, can be converted into a machine readable knowledge, machine readable data. Hence, we can understand, we can interpret, we can identify patterns from that data. So hence we can analyze genome and its structure. We can analyze a specific gene and its structure. Basically, a gene has a structure such as its UTR, untranslated regions, exons, introns. You can analyze these structures of the gene. You can analyze a protein, its sequence, its 2D structure, its 3D structure. You can design a new structure of the protein, for example, known as 3D structure prediction if there is no structure available. You can do a species to species comparison. For example, you have two bacterial species and you want to compare their sequences. You can do that using bioinformatics. If you want to identify what sort of genes are expressing in a human organism at any given moment due to any given condition, for example, disease versus control, disease versus normal conditions, you can do that through bioinformatics. If you are trying to understand what sort of mutations are occurring due to a specific disease, what sort of mutations are leading to a specific disease, what sort of mutations are leading for a bacteria to gain a new antibiotic resistance, you can do that through bioinformatics analysis. So overall, bioinformatics helps us to identify biologically important biomolecules that can help us to help with other life science research areas. For example, microbiology, zoology, botany, molecular biology, cellular biology, molecular oncology, which is one of my fields as well. So Bioinformatics overall gives you the tool, methodologies, and helps you to figure out a solution for a specific biological problem. So bioinformatics itself is not a problem that you have to solve. It is a tool overall that helps you to solve a biological question. For example, you want to understand a genome and its sequence, you can do it through bioinformatics. But the data comes from other life science fields. For example, you might know someone who is from molecular biology background and they generate data in their lab. For example, they are trying to sequence a human genome. Their job is to sequence the human genome, and your job as a bioinformatician is to take that human genome and analyze it, identify new variants, identify the gene expression, compare it to mouse, compare it to chimpanzee, for example, take a protein sequence and analyze it further for drug discovery, for 3D structure prediction, etc. So as a bioinformatician, your job is to employ the tools that you have with you and interpret the data in terms of biological knowledge. So what is not useful? So basically, most of the students, what they do is they learn these basic skills, that is sequence alignment, 3D structure prediction or 3D modeling, molecular docking, phylogenetic analysis, bioinformatics databases, and they think that they have learned everything in bioinformatics. Rather, these 5 topics that I have mentioned here specifically on the slide are those topics that don't help you in the long run. They don't even assist you with your proper projects, because you have to identify a research problem first in bioinformatics, and then you can apply these techniques that are present on this particular slide, and then you can apply these techniques onto that project and figure out a solution. But these techniques themselves do not help you in the long run. You have to understand, you have to learn new advanced skills to be able to do professional bioinformatics research. So what do I mean by that? For example, you have designed a proper project where you are trying to understand, where you are trying to develop a project where you are trying to figure out a solution to identify drug targets for a bacteria that is antibiotic resistant, and you are trying to see if this bacteria has any close relation with any other bacteria. You can do phylogenetic analysis, and once you have done phylogenetic analysis, you have identified the conserved proteins. You can then do 3D modeling, you can then do molecular docking. But learning these skills separately and then just relying on them for jobs or for other higher end research projects is not going to help you in the long run. So try and understand this, and then try and learn much more advanced skills, which I am going to discuss later on. So what are the subfields of bioinformatics? There are multiple fields to choose from in bioinformatics, but specifically the fields that are important are genomics, transcriptomics, proteomics. These are 3 core techniques. And then we have other techniques as well, other subfields of bioinformatics: metabolomics, metagenomics, and clinical informatics. These 6 subfields of bioinformatics revolve around scripting, software development, and algorithms. So none of these core advanced subfields of bioinformatics can be executed properly, can be learned properly, can be excelled properly without learning scripting, without learning software development, without learning algorithms. Yes, you can run basic tools. Yes, you can do docking using a graphical user interface, click here, click there, but they are not going to land you a higher end opportunity. They are not going to help you in the long run, because of the fact that the higher and advanced tasks only happen on Linux based computers, only happen on command line based computers, such as scripting, software development, algorithms. So you have to understand that scenario as well. We are going to discuss each of these subfields properly in the next further slides, but you have to understand that without learning scripting, you cannot do genomics projects, you cannot do NGS projects, you cannot do RNA sequencing analysis without learning scripting, basic scripting. Yes, you can do these analysis on Galaxy, but that's not how you do a professional work. Okay, so what sort of advanced technologies and techniques or methodologies are available for each of these fields that we discussed previously? So for genomics or NGS, we have variant calling. You can identify new mutations. You can identify variants that are occurring in a genome at any given moment. If you have NGS-based whole genome or whole exome datasets, for example, you are working with breast cancer and you want to identify which mutations are occurring according to a particular disease such as breast cancer, you can take whole genome, whole exome data, and you can identify variants using NGS based pipeline, which is whole exome analysis or whole genome analysis. You take your raw data sets, you map against a reference genome, you identify variants. That's it. Then we have gene expression analysis, which is also known as differential gene expression analysis, or it has another name, which is RNA sequencing analysis. So you take your data and you analyze it in a way that you are comparing one condition to the other condition just to try and find out which genes are more expressive as compared to the other set of genes that are underexpressed. Now you have proper therapeutic biomarkers available with you that can be targeted for a specific disease. For example, you are working on prostate cancer and you have a data set where you have 10 cancer samples, 10 normal samples, and you're comparing these 10 samples of the cancer against the normal just to identify which genes are overexpressive and which genes are underexpressive. So the genes that are overexpressive are known as highly upregulated genes, and then you can identify the genes that are crucial for prostate cancer according to pathway analysis, and then you can target them through proteomics, which I'm going to discuss further. Finally, is very hot topic these days, single cell genomics, single cell transcriptomics, or spatial genomics or spatial transcriptomics helps us to identify cellular level heterogeneity of each data set of each sample. For example, if you take sample from your hand and then you analyze it, single cell allows you to compare each cell to the other cell, and this is a very unique field and allows you to do cell subpopulation analysis. You can identify clusters of cells or cell populations based on a particular disease, and then you can target that specific subpopulation that is causing a disease. So on the other hand, computational drug discovery helps us to identify drug targets. It helps us to compare wild versus mutant structures. For example, a mutation has occurred in a specific protein and due to a specific disease. What you can do is you can compare the wild structure with the mutant structure and try and see what new sort of function is being gained by the mutant structure, and if it is stable over time or not. You can do pharmacomodeling that helps you to identify specific ligand compounds with specific poses that can fit at the best possible positions of a best possible protein target. You can do molecular dynamics that helps you to understand if the protein is stable over time or not using RMSD and RMSF graphs. If you have docked your ligand against the overexpressive genes, for example, and you are trying to see if your drug compound will inhibit that protein over time or not, you can do molecular dynamics to see if it stays stable or not. If it does not stay stable, that means you have designed an inhibitor and you can publish your research paper further. So another unique subfield of bioinformatics is machine learning. Machine learning can be definitely applied on bioinformatics and bioinformatics data sets. For example, if you are trying to understand where the signal peptide is in a single given protein sequence, you can do that using machine learning. If you are trying to understand the promoter regions in a biological data sets, you can do that using machine learning. But for that, you have to learn Python and R. Any of these will be sufficient for you. Disease prediction helps us to do machine learning helps us to identify disease and do disease prediction over time based on the data that we have. This comes under the health informatics fields, and then clustering and model development for machine learning, etc. So what specifically you can do in genomics? As I said earlier, in genomics, what we do is we take data from an individual or multiple individuals. The data could be whole genome or RNA based data set, and there are other data sets available too, but I'm just going to discuss these two for now. So what do we do in genomics? That we can identify potentially pathogenic variants, for example those mutations that can cause a change in the protein and eventually cause a disease. For example, if you see over here, T is changing into A, or both A's are changing into T. This is known as SNP, single nucleotide polymorphism or variant. So these are the white colored alphabets or nucleotides are the variants basically, and these variants might have a potential to change a protein sequence. Maybe these mutations are occurring in a exome rather than intronic region, so that means they might have the possibility to change the protein structure, and if they are changing protein structure, they might change its function. They might change its normal functionality in a way that it may cause a disease. So we can identify potentially pathogenic variants, we can identify rare or protein coding variants or mutations, variants with possible improve that can help us to improve livestock, crops, or do disease therapy analysis. We can identify differentially expressed genes, as I said earlier. We can do cellular heterogeneity analysis to identify cell subpopulations. We can do disease versus normal state comparison. We can do we can identify multiple species in a single individual. For example, metagenomics, you can take sample from your gut microbiome and then analyze it and find millions of different bacterial species that are living in your gut microbiome. So this is how genomics helps us to identify various new uh, various new drug targets, various new biomolecules, various new therapeutic biomarkers that can help us to solve or resolve a health issue. So how can these new skills help us to do further projects? So suppose, yes, genomics can help us to do all these different kinds of different kinds of analysis, but how do I get started? That must be your question. So basically, first of all, what you have to do is you have to choose a disease that you are interested in. Then all you have to do is follow this pipeline that we have added. Basically, all you need to do is take data from NGS based data, that can be whole genome, whole exome, or even RNA sequencing data. Then run this pipeline to identify variants, and then you have with you those variants that might have potential in a particular disease, and then you can analyze them further. So this is how you can literally take data set from any of the publicly available databases such as ArrayExpress, NCBI Geo, or EMBL in ENA. These three databases will help you to download data that is NGS based data, RNA sequencing, whole genome, whole exome, for bacterial, other species, Homo sapiens, mouse, chimpanzee, anything that you want. The data is usually available there. Just go there, search for your disease that you want to be working on. For example, diabetes, just type diabetes there in the search bar, and then you can find data sets. Then you execute this pipeline that you can see over here, but keep that in mind that you need a Linux computer for these kind of analysis. Okay, so what type of analysis can be achieved using genomics? On the left hand side, this single figure, if you see, this is a differential gene expression analysis. What we were trying to do is we were trying to compare prostate tumor versus normal prostate gland to identify which genes are overexpressive and which genes are underexpressive due to a particular specific disease. And this helps us over time because we can identify biomolecules that might be therapeutic, that might help us to develop therapeutic drug targets later down the road using proteomics techniques. But specifically in NGS, in genomics, what we do is we identify these drug targets, these biomolecules that might have potential to cause a disease. For example, if you see over here on this right hand side, AMACR or PCA3, these two genes are highly expressive in prostate tumor, and these three genes are actually being used for diagnostic purposes of prostate tumor. So what you have to do is just have to note down these genes that are highly expressive, find a gene that is novel, and then you can do further research on them. Using single cell genomics, you can identify cell subpopulations that are living in at any given tumor site or disease site. You can identify these different types of cells that are present there that might be causing a particular disease, and then you can do differential gene expression between, for example, memory CD4 cells with naive CD4 cells, and try and understand which genes are highly expressive in memory cells as compared to the naive cells, and then you can do further research on them as well. You can do pathway analysis to identify which pathways are highly upregulated, which are downregulated. You can do you can identify which genes are highly expressive in which type of cells. For example, if you see here, FOXP1 is highly expressive in plasma B cells as compared to the CD4 NKT natural killer cells. So this is how genomics helps us to identify these new biomolecules, these new therapeutic biomarkers that might help us to resolve a particular disease. So what you can do in proteomics. Over here, this is a figure that we have added from one of our research that we were conducting on a French bacteria that I cannot name at this point. But we were able to identify mutations that are occurring in a particular bacteria over time due to a drug being given, and we were trying to identify what sort of new variants can arise in a particular given species of a bacteria. So basically, this happens in a genomics project. But since we have information such as what sort of protein change is occurring in a particular protein, what we can do is we can then design a 3D model. We can predict a 3D model that can guide us. Okay, what sort of change is occurring in a given protein due to a mutation. So in proteomics, you can identify protein expression analysis. You can identify drug targets as I shown you earlier. If the protein is highly upregulated, you can identify that target. You can then compare the wild and mutant structures, as you can see in this figure, that you can compare one structure with the other structure, try and see where the new change is occurring and if the mutation is causing any difference in the overall structure of the protein. If it is causing any change in the domain region or outside the domain region, that sort of stuff can be done easily in proteomics. And this is how you can implement 3D modeling. But first of all, you have to understand a proper professional way to conduct research. You can't just rely on 3D modeling and then just start comparing mutant versus the wild. You have to understand the biological aspects of the protein as well. So you can elucidate the stability of the proteins through simulation. You can do viral genome vaccine development. You can do subterranean genomics and proteomics to identify new drug targets by singling out proteins and filtering them out and trying and find a novel drug compound. For example, we were able to identify new drug targets from a bacteria that was causing disease in cows that were in New Zealand, and this was our long-term project, and we were able to identify new drug targets, and we also developed a vaccine. So this is how proteomics helps you to cure specific diseases if you're trying to study new proteins, if you're trying to study new drug targets, etc. So if you see over here, you can dock a specific ligand against the protein. You can try and see how multiple drug compounds can interact with a single protein. Docking also occurs in microRNA and mRNA, by the way, and you can dock a specific microRNA against an mRNA, try and see how the microRNAs are regulating a specific protein, a specific mRNA's expression, because mRNAs act as regulatory inhibitors for specific mRNAs, and they also cause disease such as cancer. You can study them. For example, you are trying to design vaccines, you can then do docking between a immune-based receptor and your vaccine, trying to see if the immunogenic response can be generated from using that vaccine that you have developed or not. You can also do molecular dynamics, try and see how the protein stays stable over time or not, if it deviates from the original RMSD values or not. So what sort of programming skills are required for bioinformatics? It is the most single crucial skill. Basically, the reason is that you cannot do day-to-day tasks that are repetitive in nature. You can't do it manually. For example, you have to dock one single protein against one ligand. Okay, you can do it manually. You can run a single command. But what if you have to dock 1000 ligands against 1000 different proteins? In that case, you have to learn programming. You have to be able to create command line scripts that can automate different tasks that can be repetitive in nature. But for you, it is much more easier if you can just learn scripts. For example, if you have to name all the files that are present, name all the samples that are present in a given folder, you can't do it manually. It will take a lot of your time. So what you have to do is you have to learn some scripting to just run that code within that directory, and that can list all the files that are present there. So Python has to be learned for day-to-day scripts, or that should be utilized for advanced analysis such as RNA sequencing analysis, single cell sequencing analysis. If you want to do any kind of meaning visualizations, the visualization that you saw earlier in the genomics slide, these ones, all of them were created using Python code. So R code in RStudio. So you have to learn R for proper meaningful professional tasks such as RNA sequencing analysis, single cell sequencing analysis, or any other sort of proper statistical analysis. But if you want to do day-to-day analysis, day-to-day random scripts, then you have to learn Python, because it is much more easier to get started with Python. And Unix or Ubuntu has to be learned, because without Ubuntu, you cannot do genomics analysis. There is nothing that you can do in genomics that can be done on Windows. You have to install Ubuntu, you have to install Linux onto your computer, because it will help you to streamline your pipeline, streamline your analysis, and it is basically a safe haven for bioinformatics students and scientists. If you are not installing Ubuntu, you are cornering yourself with opportunities. You are limiting yourself with opportunities. Basically, there are a lot of opportunities that can open up if you install Ubuntu, if you try and understand new techniques such as NGS based analysis using Ubuntu, because it is high in demand and will help you with a lot of analysis as well. Okay, so what sort of analysis can be done using programming? Let's suppose the R-based visualizations I shown you earlier, and the Python-based visualizations are over here, which we were trying to understand. We were trying to understand how a specific disease can be caused due to specific glucose level or leptin level in a particular disease. I cannot name the disease right now, but we were able to do machine learning. We were able to create a machine learning model that comes under health informatics, and we did that analysis using Python. And on the other hand, if you see this figure, this is one of the pipelines that I developed for proper automated RNA sequencing analysis. And all you have to do is just give a single accession ID, and it will download all the samples for you, and it will analyze them for you automatically. So it helps me to automate my entire analysis rather than relying on one single command and running them myself one by one. So this is how scripts can help you over time. So what can you do with all these skills? I'm going to leave 6 different concepts here for you to explore. For example, you can identify differentially expressed genes for liver cancer. You can understand the structural changes that are caused due to mutations in BRCA1 protein. You can identify epitopes and pathogenic proteins to design novel vaccine. You can identify biomarker to design new drugs or execute drug repurposing. For example, those drugs that have been already tested, you can test them on your own new biomarkers that you have identified. You can develop end-to-end automated pipelines, or you can identify or study those link RNAs or non-coding RNAs that are causing some specific disease. So these are the skills, these are the concepts that I'm leaving here for you to explore further and for you to analyze further. Okay, so what about conclusion? So first of all, what you need to do for you to grow in bioinformatics, what you need to do is you need to identify and shortlist your interests first and foremost. You have to understand what do you want to do. You want to do computational drug discovery, you want to do genomics. Then shortlist your interest. Okay, specifically, I want to work on rheumatoid arthritis. I want to work on brain cancer. I want to work on skin cancer. Specify your interest. Then you have to read papers. And within papers, you have to understand the methodologies. You have to understand how and what a specific task is done. Then you have to devise a hypothesis for your research. For example, you are working with skin cancer. What exactly you want to explore in skin cancer? Each specific biological phenomena is huge in scope. You have to figure out your objectives. You have to shortlist your objectives in a way that your scope is small and you are not deviating from the original goal. You have to implement those tools that you have learned over time, command line tools, graphical user interface tools, but you should not neglect theory over time, because without relying on the theory, you cannot interpret any sort of analysis that you do in bioinformatics. Eventually, what you have to do is you have to interpret your information. For example, the plots that you have created, you have to visualize them, you have to interpret them in your research paper. So you need to know what you are doing and why you are doing a specific task. So do not enroll in courses without practice. You have to practice and you have to learn them properly. If without practice you won't proceed further, you will lose a grip on your commands, on your analysis skills over time. We are conducting an introduction to cancer therapeutics and computational drug discovery seminar and online workshop that is going to be conducted on 25th and 26th March of 2023. It is a particular workshop where you are going to learn hands-on skill to identify drug identification of targets, and then all the way to therapy development of a particular disease. So from target identification to therapy development of a particular disease such as cancer. We are not going to name that cancer right now. So introduction to cancer therapeutics and computational drug discovery. This is going to be completely hands-on workshop that is going to guide you how to take the original target, analyze it in such a way that you can develop a new therapy. We are going to do molecular dynamics, molecular docking, and all these new techniques that we are going to introduce as well. Any questions that are related to bioinformatics, you can leave them below the comment and we will try and answer them as soon as possible. If you have some specific questions, you can email us that are given under the description. If you have any specific questions related to my field, you can directly text me on LinkedIn as well. Thank you so much. Have a great day.